Welcome everyone to today's webinar. It's my great pleasure to have you all here. And I'm really happy to give this webinar on therapeutic antibody research because this is like a really important topic to me. And I hope you enjoy it as much as I will do. So this is the agenda for today. So I will first talk a bit about therapeutic antibodies in immuno-oncology in a more general way. So just a short introduction into the topic. Then I will talk about the therapeutic antibody process, which steps are involved, how is this actually working until you really get this therapeutic antibody, the final product that goes to the patient. And then also we talk about the functional screening of those antibodies because this is also like a very important step in this whole development process. So let's directly start into the topic and let's talk about therapeutic antibodies in general. So there are many different mechanisms of action how therapeutic antibodies can work on a target, or like here, of course, a tumor cell. An antibody can bind to a tumor antigen on the surface of the target cell and induce programmed cell death or apoptosis. We, of course, have other mechanisms like an antibody can bind to soluble factors that neutralizes those factors and block them from binding. Also, an antibody can bind to a receptor on the surface of the tumor cell and by that block the binding of the soluble factors to this receptor, but also activate those receptors. Then we have also the complement-dependent cytotoxicity. So an antibody can bind to the tumor antigen, and then the C1 complex of the complement system can bind against the Fc part of the antibody, what leads to the complement cascade and ultimately to the formation of the membrane attack complex, what is kind of leading to the lysis of the tumor cell. Then also we have engineered antibodies that are conjugated. So to say, it could be radiation or drugs that are conjugated to these antibodies and directly targeted to the tumor cells. We have bispecific or trispecific antibodies, so antibodies with several specificities, bridging effector and tumor cell together. Then we have checkpoint inhibitors, so antibodies that are binding against these immune checkpoints and helping the immune system to act against the tumor cells. And then we, of course, have a very important topic that's the effector cell recruitment. So an antibody is again binding against the tumor antigen, and then effector cells can bind to the Fc part of the antibody, like NK cells, inducing ADCC, antibody-dependent cellular cytotoxicity. We have macrophages that can do that they then do phagocytosis, or also T cells that are doing then T cell-dependent cellular cytotoxicity. So all these steps, or all these like mechanisms, can lead to tumor killing, and that's why therapeutic antibodies are very important in the cancer field as cancer therapeutics. And you see here also the ADCC, so this antibody-dependent cellular cytotoxicity caused by NK cells can work in different ways. So by normally it is that you have your antibody that's binding against the tumor antigen, and then the Fc receptor of the NK cell binds to the Fc part of the antibody, what then leads to the killing of the tumor cell. But this can also be done with bispecific antibodies, or also blocking antibodies are important that block the inhibitory receptors of NK cells so that they can work and kill the tumor cell. So now that was it for my introduction for the beginning. So now we'll directly hop into the topic of the whole therapeutic antibody discovery process. And this is like, has several steps, so to say. So you really start with the target discovery. So with the validation, so you really want to see like, what's the new maybe like tumor target? What can we like direct? How can we direct our therapies against this tumor? And then, of course, in the next step, after you validated your target, you do the antibody discovery because, of course, you want to have an antibody against this target. In the next step, you do the antibody production. So you produce those antibodies to be able to test them in a functionality testing, do the lead characterization. And then, of course, like antibody engineering is happening because, like, usually it can always like be a bit better. So you can always like do a bit of optimization. Of course, then you do like functional testing again if it's like really working better or not in comparison to the beginning. And then in the end, by that, you select your candidate, go into clinical testing, and then you have to have your final product that then goes into patients. So it's really a process with a lot of steps, with a lot of work involved. And we always try to make these processes like a bit more easy to digest. So we always try to put that into workflows, and that's like how we actually present our workflow, how I will present the workflow today to make it a bit easier to understand this complex processes. So we always do that in like several steps. So in our Miltenyi world with the sample preparation, cell separation, cell culture, flow cytometry, and now we also have imaging and microscopy products and instruments that also help you with your work. And so we have a lot of products you see here on the bottom. So I will try to explain like the workflows in this kind of way. So let's directly start into the target discovery and validation because this is like also like a very important step in this whole process because when you don't have the right target, it's really like complicated to work. So this you can do with this Maxima imaging platform. That's a ultra high content imaging system where you can analyze like really hundreds of markers with a single sample. So it's really like an automated sampling process and imaging process. And you can image like all types of fixed samples from tissue to actually single cells. And here on the left hand side, you really see this like nice picture and how it looks like. So you can really see your tumor cells and which cells are surrounding the tumor. Where are actually like the B cells located? Where are the NK cells? Where are the T cells? So it's really interesting. And with this, you can also find out new targets because you really see on your tumor sample where are actually our tumor antigen. And so here we did this for a glioblastoma tumor sample, and we actually found out that actually it is just a restricted expression of this established markers in this tumor. So you see this like here and here. So there's actually not so much expression of this like commonly known and used markers that are also used for CAR-T therapy. And we actually identified this like, 4 new markers, A, B, C, and D, that have higher expression levels, as you see here on the left, and also higher percentage of positive cells. And you also see this like here in green with like 2 glioblastoma samples, so that really like the sample looks like really green in all the sample in comparison to like this like commonly established markers. So it's really a nice way on how you can really like look into your tumor sample and see what's actually going on in there. So that was it like so far from this like side step. Let's now talk about how you actually discover antibodies. How are you getting your lead candidate? And now we're hopping into like my first workflow. So of course, like you start with here the sample preparation because you want to have your antibodies for maybe like a human cancer patient, recovered patients, maybe like a mouse model and want to get the tumor out, the spleen out, or maybe you're even like starting with blood products. You do your sample processing to get single cells and then you separate your B cells. And of course, you want to see like, do I really got nice B cells and do nice QC analysis, do flow analysis, cultivate your B cells because they're not like usually not really frequent, especially when they're coming from tumor samples. And then you can select in the next step for the binder. So like really your antigen, your target positive B cells. go into expression of your antibody, your lead candidate, and really go then into the functional testing. But actually, with this workflow, with this process, there are coming a lot of challenges for you. So it can be that you have many different kits that you can use for the B cell separation, and this is really confusing sometimes. Also, cell activation, you don't want to get activated B cells after your B cell separation. And also, when you do single cell sequencing, the RNA quality is really important. But no worries, we have a solution for all these challenges. So I put together for you this slide. So this is we have some nice materials that show you an overview of the B cell products, really from which starting material can you get, which subset of interest, and which kit can you use. Also, here I wanted to show you that cell activation with the B cell separation is not happening. So we couldn't find any upregulation of activation markers in positive or in negative isolation. So here we have the pan B cell isolation kit, and you see after separation, the unstimulated cells are not activated. Also with the positive separation, it's looking the same. Just when you add external stimuli like CD40 ligand and IL-4, your B cells are getting nicely activated as they should. Another step and another complicated step for the B cells is the B cell cultivation, because this is not so easy. It's really time consuming. Primary B cells are not easy to cultivate. Sometimes they just die, they're not growing, and the expansion is not sufficient. So that is sometimes a bit complicated. And that's why we developed this nice human B cell expansion kit that has a standardized 10-fold expansion of your B cells in vitro within 14 days. It preserves all your B cell subsets, and it's really a standardized and efficient activation of B cells. If you want to know more about this, we made a very nice scientific poster about this topic, tested different settings, different cytokines, and checked for the subsets and so on. So if you want to know more about this, when you get the PDF, just click here and then you get more information on that. Then the next step would be the binder selection. So you really want to have your target positive, your antigen positive B cells. And this is a bit tricky because it's a very rare subset. B cells are very sensitive. You don't want to have any contaminations or sample carryover. And also it's high safety level acquired when you are working with infectious materials, so depending on which patients you are having. And there we have a very nice solution that's the Max Quantito. So it's a very gentle microchip-based sorting process. What's especially advantages for this very sensitive B cells. It's in a closed and sterile cartridge system, so you have no contamination, no sample carryover, and also very safe for operators because you have this closed cartridge. You do flow sorting based on up to 10 parameters, where you can also really nicely sort B cell subpopulations. Here's just an example of total B cells. So here you see your B cells before sorting and here after sorting, what worked really nicely. So another challenge is always the cell analysis. So this is always a problem of background, unspecific binding, the reproducibility, and that it always takes so long. And if you want to know more about this, please watch our webinar that we hold in June. So there's of course a recording available. So when you just later here click on this link, then you can just watch the webinar where we explain all the solutions to these challenges. And now I come to a conclusion of this workflow, so to say. So the kits that I would recommend you to use in this workflow, it's tumor dissociation kits and the Gentamax to do your sample preparation. For the isolation of B cells, straight from kits where you can really directly isolate your B cells from blood products in a really fast and also possible automated manner that really saves you a lot of time. But also, like, you can, of course, do isolation of B cells from PBMCs with PAM B cell isolation kit, or also CD19 microbeads. Also do cell analysis with free affinity antibodies and also our MaxQuant analyzer. And for the B cell culture, I really recommend you our nice B cell expansion kit. We also have CD40 ligand multi-mag kits for human, TLR ligands, and a lot of cytokines that help you with B cell culture. For the binder selection, I recommend you to use the MaxQuant Title, and then you're almost done and go to then expression and functional testing of these antibodies. So now we have our lead candidates. Okay. So now we want to know what are they actually doing. So now functional testing is happening for this like lead characterization, but also for the optimization because you really want to make them work better. And for this, we have tons of different assays. So I just selected some of them and just put them here on the slide so that you see there are like so many different things that like, an antibody can do and that are tested, but I cannot walk you through all this like workflows and all these assays today. It would take like, really, it would get really, really late. So I just focused on two of them. So as I like you mentioned before, the ADCC, so the antibody-dependent cellular cytotoxicity assay, and the mixed lymphocyte reaction that's also used a lot when people are screening checkpoint inhibitors, and that's why I choose that one. So let's directly hop into the topic of the ADCC. So the workflow looks like this. So you also get your blood sample here in case, like, of course, of a human, and isolate your NK cells. You expand your NK cells, and then you go into the co-culture of your antigen-positive cancer cells with the NK cells and your antibody candidate up here. But you also do the same without the antibodies to be able to, like compare it. Where is killing happening and where not? And then, of course, afterwards, you have the readout with the flow cytometry and really check for annexine or PI to really see where is the killing happening. So what is going on there? What does the antibody do? Challenges can be here. Also, of course, cell activation and cell separation, that you sometimes have like really low volumes of blood because you have like, patients or clinical samples maybe. So there's sometimes maybe not like so much. Then the NKT cell contamination in that sample that is possible. And that like, automation because like people are doing like tons of this assays, several of these assays, not only the ADCCs one, maybe also like, all of the other ones that I showed you before. So automation is like really important here. So, and I just wanted to show you here that all this is solved without our kids. So with the straight from Buffy code CD56 microbeads, you can isolate your NK cells directly from blood, what saves you a lot of time and can also be done in an automated manner. But of course, you can also use the NK cell isolation kit, so isolate your NK cells from PBMCs and also do that in an automated way. In both cases, you get nicely isolated, non-activated NK cells. Of course, with the CD56 microbeads, you also get the NKT cells, but it doesn't really matter in this assay because they have no FCR receptor. So NKT cells do not express CD16, so no antibody binding can happening, and so they do not interfere in this assay. So it really doesn't matter. And I show you the data here. So here's the comparison of this natural cytotoxicity in comparison to ADCC with our cells isolated with both kits, so with the straight from kits and untouched. And the killing is really happening nicely and just like looks the same no matter if you're doing straight from or untouched. And here we just like use different ratios of cells. So if you want to know more about the ADCC assay, please later click in the PDF on this. Here we have a nice application note what explains all the details and all the data that you need to know about ADCC. And these are the products I recommend you for this assay. So I told you before, the straight from microbeads to isolate your NK cells from whole blood, Buffy-coat, Leukopak, or LRSC. Then also the NK cell isolation kits with the isolation of NK cells from PBMCs. Then the NK max medium and max cytokines. And here the NK max medium is like really great because it helps you that your NK cells are not getting exhausted, because that's sometimes what happens at NK cell culture. But with the NK max medium, this is really not happening. And then for the readout, you of course have a lot of reaffinity antibodies and the max quant analyzer, where you can analyze the readout of your killing assays in the ADCC. And with the max quant X, this can also be done in an automated way, but really nicely. And now we come into the second assay. So now we're talking about the mixed lymphocyte reaction. What I told you is more done for the checkpoint inhibitors. So this is an assay that's like sometimes really a bit, confusing because it can be done in many different ways. And that's why it's sometimes a bit that people are saying MLR to like many different things. And so we put together here are 3 options that you can do. So option 1 is, so you're getting your PBMCs from donor 1 and mix them with PBMCs from donor 2. But you can also do option 2, mix the T cells from donor 1 with the antigen presenting cells from donor 2. But you can also do an MLR, it's also an MLR when you mix just like the T cells of donor 1 and donor 2. You mix them all together here in the tissue culture, add then your lead candidate, your antibody, checkpoint inhibitor, and then you look for proliferation and activation. Here now I will focus on option 2, so mixing the T cells with the antigen presenting cells. The workflow looks like this. So you have donor number 1 and donor number 2 and go into monocyte and T cell isolation. And for your monocytes, you need to differentiate them into monocyte-derived dendritic cells, so to say modices, in culture, and then you have your antigen-presenting cells, your modices. You mix then the modices with your T cells in culture, add your lead candidate, your antibody, and then you're going directly after a bit of culture into the proliferation assay and check in the flow for activation and proliferation. Challenges can be here that sometimes the modices have a suboptimal antigen stimulation. Inconsistent cell culture results is sometimes always like a problem also here with the modices, and also difficult translation into clinics. But don't worry, I have also like a solution here. So with the max premium you get cytokines, you can really optimally differentiate and maturate your modices. So we could really show that they are getting into a mature phenotype, that their migration capacity is really working nicely, and also that they produce IL-12. So really all the criteria that you need for your modices. And with this exact unit per microgram dosing that we have with our premium grade cytokines, you always stimulate your modices in the right way. So you're really doing always using the same activity, so to say. So you're not putting in like one time much more than the other time. So it's like always like really standardized and always the same. So here you see some example data, how MLR is looking like. And if you're interested in like more details and more data, more explanation, just like click later here on this application note that we did on the MLR assays. The products I would recommend you here are also the straight from products. So we have them for B cells, we have them for T cells, we have them for NK cells, and also for the monocytes, so CD14 microbeads, CD4, CD8 microbeads. So also again, directly getting your cells from whole blood, from Buffycomb, from LRSC, and from leukopak, what saves you a lot of time and can be automated. Then, of course, we have all these kits where you can isolate your cells from PBMCs. So CD4, CD8, CD3, CD14 microbeads, and also the untouched kits for T cell isolation and monocyte isolation kits. So you can really select here what fits your project, what fits your needs the best to select really here the product that you want to have. And then in the step of cultivation and modus C differentiation, you can use the max premium gate cytokines IL4 and GM-CSF to really differentiate your modices. For the T cells, I would recommend you the Tex-Mex medium. That is really a great medium for T cells. What gives you high expansion of T cells. What's really nice. And for the readout, of course, the reaffinity antibodies have all you need to really get your proliferation analysis done. And with the max quant analyzer, you have the nice readout. And as I told you before, with the max quant X, this can also be done in a very nice automated way, so that this is really done while you maybe go back to your office and drink a cup of coffee or listen to a nice webinar that we are doing. So in summary, I just wanted to tell you, and I hope I made it clear to you, that immuno-oncology assays are really important tools for the development, assessment of novel therapeutic antibody candidates. And we really support you with the entire workflow of the therapeutic antibody discovery process with simple, standardized, optimized, and automated solutions. And not only for these assays and applications I showed you here, so there's much more. And I really hope that I could help you and that we can help you to fight such diseases as cancer and, of course, also infectious diseases as SARS-CoV-2. And now I thank you very much. So I have here the disclaimer, and I thank you very much for your attention.